Welcome to our video lecture on topic 2.1, Molecules to Metabolism. Our essential understanding, the big idea that IB wants us to come away with, is that living organisms control their composition by a complex web of chemical reactions. That's basically what metabolism is, and it's one of our objectives for today's lesson. We are going to define metabolism. We're going to also talk about carbon, some of the chemistry of carbon and why it's the basis of living things. We're going to describe the four categories of biomolecules. We have carbohydrates and lipids and proteins and nucleic acids. We're going to also contrast the two sides of metabolism, anabolism and catabolism. Our essential understanding, again, is that living organisms control their composition by a complex web of chemical reactions. That's what metabolism is. Molecular biology or biochemistry is the science of explaining living processes, so nutrition and growth and homeostasis and reproduction, in terms of the molecules involved. So we're going to focus very much on all those biomolecules, lots of chemistry. How organic chemistry started was with the synthesis of urea, and urea is one of the components in our urine. And a long time ago, we had this idea of vitalism, that only living things could make stuff like urea, that only organisms could make organic compounds, that we had some amazing vital force that made us magical, more magical than a laboratory could ever be. Kind of all based back on Aristotle's idea of the psyche, that our breath, our soul, was what allowed us to be alive, and it didn't have much to do with chemistry. But then this guy, Friedrich Roller, he actually was able to make urea in the laboratory. He made this molecule of urea, not with his liver, not with a dog kidney, but instead in the lab. And this was the beginning of organic chemistry, biochemistry, where we could, in the laboratory, make and analyze these compounds that had been previously thought to be made only by living things. All of these molecules that us living things make are based on the element carbon. Carbon is the 15th most abundant element on the planet, but it is definitely the most important to us, us living things. Carbon makes covalent bonds. Remember that covalent bonds are bonds where electrons are shared. And what's pretty cool about carbon is that it can make 4 covalent bonds because it has 4 valence electrons. Remember that valence electrons are the electrons in the outermost energy level. This energy level would not be considered the outermost. It's the inner energy level. This energy level, however, is carbon's valence energy level, and notice that there are 1, 2, 3, 4 electrons, a valence of 4 on carbon. This means that to get to the octet rule, remember that octet means that atoms are most stable if they have 8 electrons in their outermost energy level. So for carbon to get to that octet rule, to fulfill the octet rule, it needs 4 more electrons, because 4 and 4 makes 8. So those 4 covalent bonds can actually be single or double or triple bonds. Carbon can bond to other carbons or to different elements in lots and lots of ways. And that flexibility of carbon to make so many bonds is what gives it the versatility to be the basis of living organisms. Carbon is found in oh so many molecules. This is called ethanoic acid. We know it better as acetic acid or vinegar. Notice the carbons in there. This is a saturated fatty acid, super important for our cell membranes and for some energy storage. Our molecule here has, oh my goodness, so many carbons. Those carbon chains just go on forever. Unsaturated fatty acids, these tend to be more oils. These tend to be more fats, the saturated guys. And so notice that there's a double bond between the carbons. It makes this little bend in the molecule. Unsaturated fatty acids versus saturated fatty acids. This guy down here is ATP, adenosine triphosphate. And what's kind of interesting about this guy, you might notice there are no carbons. And you're like, wait, how is this guy on a slide about carbon if there aren't any carbons? Well, organic chemists, biochemists are sometimes a little bit lazy and they don't draw the carbons because it is assumed that there are lots of carbons. And so all these points in the molecule where we have some kind of intersection, but no atom labeled, these are all carbons. So there are actually lots and lots and lots of carbons. We just didn't label all the carbons because we know that there are carbons in the molecule. There are 4 main groups of biomolecules, and they all have carbon in them: carbohydrates, lipids, proteins, nucleic acids. We're going to talk about all of them very briefly today, and then in a later lecture, we're going to go into a lot more detail. Not one later lecture, lots of lectures on each of them. First up, let's talk about carbohydrates. So carbohydrates are composed of carbon, of course, and some hydrogen and oxygen. What's pretty cool about the hydrogen and oxygen is that they are in a 2-to-1 ratio, just like water is in a hydrogen to oxygen ratio of 2-to-1. And this is why we call carbohydrates carbo for the carbon, hydrate for that 2-to-1 hydrogen to oxygen water-like ratio. The functions of carbohydrates are energy storage and structure. This is glucose. Glucose is what we call a monomer. It's a very small piece of carbohydrate. This guy over here is a polymer. It's a chunk with many, many little pieces stuck together. Those pieces actually happen to be glucose. So each one of these little chunks is a glucose, 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 and they're all bound together, to make this polymer, and this polymer happens to be called glycogen. Glycogen is a carbohydrate storage molecule in animals. It's basically the animal version of starch. So starch is plants, glycogen is for animals. Lipids are pretty similar to those carbohydrates. They have carbon and hydrogen and oxygen also, but they do not have hydrogen and oxygen in a 2-to-1 ratio. Instead, there's lots of carbon and lots of hydrogen and very, very little oxygen. So notice in this molecule here, we have some oxygen, but it is nowhere near the numbers of carbons and hydrogen. So very little oxygen compared to the carbon and hydrogen in our lipids. Lipids are fats and oils. Fats tend to be solid at room temperature, oils are liquid at room temperature. Both of these guys are insoluble in water. They do not dissolve in water very well. We use lipids for energy storage. Fat, or the nicer scientific word is adipose tissue, is our energy storage. Also homeostasis. This molecule is testosterone. That we need testosterone. Sorry, I got lost in my spelling there. That we need for homeostasis, one of the hormones that helps us maintain homeostasis, those internal balances in our bodies. And then also structure. And this molecule is a phospholipid. Because it has that phosphate group and then it has those fatty acid chains. This is what makes up our cell membranes, the structure of our cell membranes. Proteins up next. So again, carbon and hydrogen and oxygen, it's a theme. They're everywhere. But now we're going to add in lots of nitrogen and sometimes a little bit of sulfur. Proteins are composed of amino acid chains. This is what we call a generalized amino acid. All amino acids have all of these pieces in common. And then this side chain, we sometimes call it the radical group, is going to be different from one amino acid to the other. There are about 20 amino acids that we use to build our proteins. Some people would argue that this may be 21 or 22 because some of the amino acids can have some variations, but most of the time we go with 20-ish. It's close enough. We use our proteins for structure. So our skin, our hair, collagen and keratin is very much a protein structure. And then metabolism, our enzymes. And we'll talk a lot about enzymes in our second unit. Our enzymes are going to be those molecules of metabolism. They're going to make those chemical reactions happen. Lots of different kinds of amino acids have different properties, and by putting these different amino acids into different orders in our proteins, we are able to make proteins with these different properties and therefore these different functions. So we have some that are nonpolar. Nonpolar means that they don't have charges. They do not dissolve very well in water at all. Some of them are polar, where they have like a positive side and a negative side. This does allow them to dissolve a little bit more easily in water. And then we have some that are more than polar, but they are actually charged. So they might have a positive charge, they might have a negative charge. These guys, again, a little bit more ionic, allow the protein to have that charge to move around through the water a little bit more differently than those non-polar amino acids will allow. Last up, our last category are nucleic acids. We're talking about DNA and RNA. The NA in DNA and RNA stands for nucleic acid. These guys are made up of, once again, carbon, hydrogen, and oxygen. We're going to throw in some nitrogen like we did with the proteins, but now we're adding in phosphorus. Phosphorus is only in nucleic acids, and that sulfur was only in protein. The job of nucleic acids is to pass hereditary information onto offspring. Nucleic acids, these nucleotides, a nucleotide is the small piece of a nucleic acid. They have 3 chunks. There's a phosphate group. There is a 5-carbon sugar that we draw as a pentagon, and then there's going to be some nitrogenous base, and this nitrogenous base happens to be adenosine. And so this would be the A, the adenine in A's, T's, C's, and G's of DNA. We sometimes abbreviate our nucleic acids, our nucleotides, with just the the phosphate group as a circle, the sugar as that pentagon, and then a rectangle to show the nitrogenous base. But those 3 pieces, phosphate, sugar, and nitrogenous base, super important to our nucleic acids. Let's review. Tell me what we see here. This guy has carbon, 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 carbon. Notice that we've got quite a bit of oxygen here. This is a carbohydrate, and it happens to be ribose, which is the sugar that we use to build RNA. This guy, notice that I've got carbon, 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 just a little bit of oxygen, not quite as much oxygen as carbon. This one with this double O and then an OH at the end makes it a fatty acid. It's a lipid. So lots of carbon and just a little bit of oxygen makes this guy a lipid. It happens to be a fatty acid. This fatty acid actually happens to be butanoic acid, which we find in butter. This guy, notice that I've got carbon, carbon, another carbon. We have that double OOH, just like we had on the fatty acid, but now I've got a nitrogen thrown in here, so it can't be a lipid because of that nitrogen. This guy is actually an amino acid. This amino acid happens to be alanine. You don't need to know that, but it's an amino acid that's going to build up a protein. Last up, this guy, notice that I've got a phosphate group. We've got that 1, 2, 3, 4, 5 carbon sugar, and then I've got some stuff with nitrogen in it. Because it's got phosphorus, it's going to be, of course, a nucleotide. This one happens to be thymine. So kind of cool. You don't have to know that, but you do need to know that it's a piece of DNA. All right, so now we're going to play Name That Molecule. What do you think this guy is? Is it a carbohydrate? Is it a lipid? Is it a protein? Is it a nucleic acid? Notice that we've got those carbons here. We've got the double OOH like we have in lipids, but there's a nitrogen. So hopefully you are thinking, haha, this must be protein. And you'd be right. This is in fact what we call a dipeptide. And this is a dipeptide because there are actually 2 amino acids bound together. We've got one here, and then we've got the other one here. These both happen to be glycine, so you would call this glycylglycine, kind of a funny name. But it is a dipeptide because it is formed from 2 amino acids, and we know that it is protein peptide because of that double OOH and because of the nitrogens, but notice there are no phosphoruses, no phosphorus atoms or phosphate groups, so it can't be DNA. How about this guy? What do we think this one is? We've got 1, 2, 3, 4, 5, 6 carbons. We've got 1, 2, 3, 4, 5, 6 oxygens, lots of hydrogens. This is going to be a carbohydrate, and this carbohydrate happens to be glucose. How about this one? Oh my goodness, crazy sauce. So all of these guys that aren't labeled are carbons, and those carbons all have hydrogens to get to their valence of 4. What do we think? What do we think? Definitely a lipid, right? So many carbons, lots of hydrogen that we can't see, but we have that double OOH at the end, but we don't have any nitrogen, so it can't be an amino acid protein. So this is what we call a fatty acid, and this one happens to be unsaturated because it has that double bond between those 2 carbon atoms. What's next? This guy. Notice I've got a phosphate group. We've got this 1, 2, 3, 4, 5 carbon sugar, and then some nitrogen up here. That makes this a nucleotide. And this nucleotide happens to be cytosine. Kind of cool, right? You don't have to know that it's cytosine. You do need to know that it's a nucleotide. All right, so let's go back to our main idea for today. Our essential understanding is that metabolism is the web of all enzyme catalyzed reactions in an organism. So us living things, these processes happen because of this web of chemical reactions. This map that looks kind of like a subway map from New York City is actually a map of lots of chemical reactions. And so we can see that these chemical reactions are turning molecules into other molecules. We add some hydrogen, we get some phosphate groups. We have some more phosphate groups popping off. We've got some CMP and some ATP and some NADPH. So all of these pieces are just some, a very, very few of the pieces that are happening in the liver. Crazy sauce. Lots of enzymes, lots of reactions, lots of chemistry happening in our liver all the time. We will not go this far into detail of metabolism, but we do need to know that metabolism comes in 2 varieties. The 2 varieties of metabolism are anabolism and catabolism. So anabolism, we're going to make some complex molecules from simpler ones. You can remember that anabolic steroids are bad for you and they build muscle, but they also are bad for you. They hurt your heart, they hurt your reproductive system. So don't do them, even though they do help to build muscle. So building things up like anabolic steroids, anabolism. We're going to use some energy to build new bonds between molecules. We're going to take these monomers and we're going to glue them together to make that polymer. And we're going to call this kind of chemical reaction condensation. We'll talk about why we call it condensation on the next slide. Catabolism is the opposite. Catabolism, we're going to cut things into smaller pieces. We're going to digest those complex molecules to form simpler ones. What's interesting is that energy is released when we break those bonds. So instead of adding energy endergonic, like anabolism, it is now exergonic energy is released. We're going to do this catabolism with some hydrolysis reactions. Again, we'll look at that a little bit more closely in 2 slides. So this idea of anabolism, where we're building things up, we're making bigger molecules, which requires energy, happens via condensation reactions. Why do we call these condensation reactions? Let me show you. So here we're going to take 2 pieces of sugar. This happens to be glucose and glucose again. And what we're going to do is we're actually going to take these 2, we call these hydroxyl groups. So these hydroxyl groups, these OHs, we're going to pull the OH off of one of them and the H off of the other guy. And when I put 2 H's and an O together, we make water. And so we call this a condensation reaction. Sometimes we call it dehydration synthesis. But it's called this because we're literally pulling water out of the chemical reaction. So we're producing water, we're pulling water out from between these 2 molecules when we glue them together. The bond that is left is going to keep just one of the oxygens, and this one, because it is between pieces of carbohydrate, we're going to call it a glycosidic linkage. It is a covalent bond. We are sharing electrons, and it was produced, this bond was produced by a condensation reaction because we pulled out that water and left some sticky ends where those 2 pieces could get glued together. The opposite of anabolism is catabolism. Catabolism literally is going to be the opposite. So instead of building things up, we're breaking things down. It's decomposition or digestion. Instead of using energy to build a new bond, we're going to release the energy by breaking a bond. And the opposite of condensation reaction is hydrolysis reaction. We're going to take that bond that we built by pulling the water out. We're actually going to add some water back in. So I'm going to take this water, which is hydrogen, oxygen, hydrogen. I'm going to break this bond. I'm going to put these guys here. I'm going to put this guy here, and we end up with two separate pieces. So the OH from water ends up on this side, and the H sticks onto that O to make the OH that's over here. And so we can add the water to split that molecule, which is why we call it hydrolysis, hydro water, lysis, splitting or breaking. Hydrolysis is the opposite of condensation. And my friends, we have accomplished video number 1. We talked about what metabolism is. Metabolism is the sum of all enzyme catalyzed chemical reactions in a system. It is the complex web of chemical reactions that keep us alive. We talked about the valence of carbon. Carbon has 4 valence electrons, which allows it to make a lot of diverse bonds, which allows it to do all the things that we need it to do to stay alive. We talked about those 4 categories of biomolecules. We have carbs, we have lipids, we have proteins, we have nucleic acids. And we wrapped up with that contrast of anabolism, building molecules up with condensation reactions, catabolism, cutting things down with hydrolysis reactions. Good work today.